Okay, so this is going to be our second part of our audio lectures for chapter 20. So this audio lecture is going to focus on stem cells, so tissue renewal, and then cancer, a defect in tissue renewal. So we want to keep in mind that many cells in our body are under constant renewal due to just how they function, how cells are made. We're going to constantly have to be making new cells. So, our red blood cells wear out, our immune cells need to be created and replenished. Also, epithelial, epidermis cells like our skin, those are going to slough off, and so we need to constantly be renewing those. And so how that is achieved is by having stem cells, and those stem cells actually will self-renew. So a stem cell would split and divide, but some of those cells are retained as stem cells or even progenitor cells, and then some of them actually then get terminally differentiated into the cell type that is needed. Okay, so there's a lot that goes into going from a stem cell to a terminally differentiated cell, and that's a vast area of research in pretty much any cell type or organ system. Is how do you take a stem cell and push it through the lineage in order to get this terminally differentiated cell? What are the different factors? What are the different signals that need to be activated, and at what time point does that need to be done? And so in most cases, when there are embryonic stem cells that are able to generate any cell in the body, but we also have stem cells that are differentiated that they can only become certain tissues or certain cells within a tissue. So we'll have these progenitors that are kind of committed to a certain lineage or a certain cell type, but even just getting from a progenitor cell to the terminally differentiated cell is quite the feat, because there are certain factors and cytokines and signals that have to be given to the cell in order to get to that step. And so again, there's researchers who are looking at this to push progenitor cells into certain types of immune cells, so that if you are deficient in an immune cell, we could give you those immune cells, for skin grafts, instead of relying upon donors for skin grafts or other types of skin grafts. So one type that's used is like fish skin, is been actually quite successful in doing skin grafts. But what if we could take your own cells and actually make skin for you? And so again, these become these holy grail type research projects that researchers work on. So as I mentioned, the reality is that a stem cell will divide. Some of those stem cells will stay undifferentiated so that you have this renewal population. But then other ones will then be committed to like a progenitor. And then eventually, as they grow and divide, some of them become differentiated into different cell types or into the same cell types. And so this allows for the tissue not to become overpopulated by a single cell type, and so that we have these progenitors that are ready to go, that aren't going to take as much time to differentiate into the terminal differentiated cells as if we had to go back to the stem cell population. So one tissue that is a good example of tissue renewal is in the gut. As we've mentioned, with digesting and absorbing, with material moving through, there is a need in order to renew that tissue. So there's these changes in pH that put the tissue at risk of losing some of those material. We also have lots of specialized cells. So we have cells that are specialized in absorption. There's cells that are specialized in secretion of mucus, secretion of enter endocrine signals, digestive enzymes, specialize in receptor signaling. And so by having these progenitor cells, this allows for all of these different cell types to originate from the tissue. And it allows for the epithelial lining to actually have this turnover of less than a week. So if we look at our figure on the right, up at the top, we can see the structure of our gut, which we've already talked about in our first part of our lecture when we were talking about the epithelial cells. But we can see how we have these villi that are formed, and then these crypts that are present, these of the epithelial layers, and then we have the connective tissue underneath. So if we look at these crypts, which is on the bottom of this figure, we can see we have this area where we have these gray cells that are non-dividing, and they're differentiated into these pamphlet cells. And then just above that, we have these cells that are colored in red. These are going to be our stem cells. So these are going to be slowly dividing, so their cycle time is over 24 hours. So if we remember back when we were talking about the cell cycle for chapter 18, that we had said that cells are going to have a certain cycle in time in which they double. And so 24 hours that we would recognize, greater than 24 hours would be slowly dividing. And then just above that, we have the yellow cells that are colored in yellow. And these are going to be our rapidly dividing cells. So these originated from the stem cells, and they've slightly differentiated into progenitor cells. And then just above those, we have our blue cells. These are not dividing anymore. They have differentiated, so these cells would be in interphase, right? And so they've differentiated into the different cell types. So it's in this blue layers where we would see the different cell types that we were talking about, like the absorptive cells that are going to absorb nutrients, like the goblet cells that are going to produce cytokines, our enteroendocrine cells, and even our tough cells. Okay. So that as we move up to the top of the villi, this is where we're going to see most of the cell loss. So this is where cells might be sloughed off and shedded, or they might undergo apoptosis, controlled cell death, so that as these cells are being renewed from the base of the crypt and moving up, it's not that the villi is growing larger, that you're actually losing them at the same rate in which you're adding them. And so generally the transit time is about 3 to 5 days from the crypt up to the top of the villi. So again, this corresponds to this about a week of turnover for your intestinal lining. So again, think about that, like every week you have a new gut. So obviously if there are imbalances in this turnover time, that could lead to the loss of the villi. Obviously, if there's things like inflammation going on, if there's a parasitic infection, immune response, that would impair this ability for the tissue to renew itself. And so generally what you would see is a shrinking of the microvilli, which then affects absorption. Obviously, it affects other aspects too, but clinically, the lack of absorptions of nutrients, because we don't have nice villi structures, is one of the pronounced clinical aspects of that. So again, I'm just giving you an example of how clinically we can see if tissues are not being renewed. So as I mentioned, we have these different types of cells that are going to make up our intestine, and so we need to make sure that we have these different cell types being repopulated as they are being lost. So to link this in with our cell signaling pathways, there are different pathways that are important. So depending upon whether these pathways are activated is how you end up with your different lineage, your different cell types. And so with Wnt pathway activated, cells will begin to proliferate. So as we are increasing the number of cells, that starts to move the cells up towards the villi. And then when the Wnt pathway gets deactivated, this causes the lack of cell proliferation. So at the top of the villi, Wnt pathway would be inactive in that blue area, so that we end up getting these differentiated cells that are no longer proliferating, that are in interphase, that are providing their function. So then we're looking at the differentiation between our different cell types, having cells that are absorbed versus cells that secrete. We can look at the Notch pathway. And so when the Notch pathway gets activated, that leads to the stem cell becoming an absorptive cell, where if we do not have Notch being activated, and we actually have delta signaling being activated, that allows us to have a secretory cell, such as the goblet cells that are going to secrete mucus. So as I mentioned, there's embryonic stem cells that can become any cell in the body, but then there's also adult or human stem cells. Adult and human stem cells generally can only become the cells that make up the tissue that they're generated from. So an embryonic stem cell hasn't undergone any kind of differentiation, but adult stem cells have, and again, it's usually just the cells of the tissue in which they can become. So in culture, we can actually keep adult or embryonic stem cells growing indefinitely, and so if we give them LIF, leukemia inhibitory factor, this actually keeps them in their stem cell state indefinitely. So we could then take those cultured embryonic stem cells, and then depending upon what signaling molecules, what growth factors, cytokines we put into the media, we can differentiate them into specific cell types. Now, again, this is a little bit of a holy grail, so definitely researchers have made progress on being able to do this, but it's not that we're able to make every single cell type. In a lot of cases, it's more work is being done on the human adult stem cells and being able to differentiate them into the different cell types that they're from the tissue that they're isolated from. So again, with stem cells, they have that potential to differentiate into entire tissues or organs, and so that we could take these cultured embryonic stem cells and that they would actually be able to form like a retina, right? So that a lot of those developmental processes that would happen in vivo in an embryo could happen in a tissue culture dish without having a whole organism present. Obviously, if you had the hindsight to freeze down embryonic stem cells from yourself or from your children, you could use those to grow tissues or organs if your child or you needed them in the future. Again, the issue is most of us don't have the resources in order to do that, or this was not technology available when I was a child, or we could do embryonic stem cells when my children were born, but again, the cost is prohibitive for most people, and so this is where more of the research is looking at adult stem cells and taking them and pushing them. And again, research has been done where they can do that. Some of the research, if you've seen the research, there was like one that came out about 5-10 years ago that had this mouse that had an ear on its back. There was an awful lot of scaffolding that was done in order for it to be a fully developed ear. So it wasn't that they grew an ear out the back of the mouse. So we just want to keep in mind that if we see something about this in the media, or that we are going to the actual source and reading the primary literature to understand that culture, but what was done in order to get to that point. And there's a lot of steps that are involved in that process. And so for the average lab, the average person, again, it becomes cost prohibitive. So at this point, this is where I would advocate to be a tissue organ donor, because that is where we're currently in our medical knowledge in doing organ transplants or tissue transplants. We are not at the point where we can be growing livers for everybody who needs a liver, or growing a kidney for everyone who needs a kidney. And those are pretty complex tissues, and so we're not there yet. Simpler tissues like retina or skin, we are at that point, but again, not to the point that it's mainstream, that anybody could walk in and be like, I want a skin graft that's my own. But maybe someday we will be there. Okay. So next we're going to look at cancer. So we can think of cancer as these defects in tissue renewal, defects in apoptosis. And so again, we'll talk about reasons where cancer happens. So for this figure is just showing some different ways in which cancer can be detected. So, through x-rays or CAT scans, in some cases, large tumors can be detected. Usually if we're doing a biopsy, if it's a large enough tumor, we might be able to see that tumor on its own. In smaller tumors, in some cases, we would do a biopsy and then do histology. So our bottom left figure is a histology section of a biopsy, and you can see how we have this tumor located in the middle. And so you can see that there's a difference in the tissue around it. It has a visual difference that we can recognize that. In some cases, especially if we're talking about precancerous cells, a histologist who is trained and certified can actually look at changes in cellular structures, and they're looking for different effects that happen when you have a cancer cell present. So sometimes cells can fuse together, sometimes they'll be multinucleated, and so these are not normal in cells, and so these abnormalities would indicate that you either have a precancerous cell and that maybe treatment would be warranted. Okay. So we want to keep in mind that cancer occurs when cells break the rules. So normal growth and homeostasis in a multicellular organism is that a cell grows and divides, differentiates, you get tumor, cells stop dividing. With homeostasis, there's a balance between proliferation of cells and loss of cells, so cell division and apoptosis. And so with a tumor, there's an excess of cell division and there's a loss of apoptosis. So you don't get that cell loss, so that balance at homeostasis is off. So some of the terms that are used when we're talking about tumors, a benign tumor would be one that's non-invasive, so it's not metastasized. It's staying in the location that it is, but we do have a increased amount of cell division present. With a tumor that is malignant, this would be recognized as cancerous. This is where it started to invade the surrounding tissue. So it's gone into the connective tissue layer or other tissue layers that are present within that tissue and organ. And then we can have metastasis, and this is where the cancer cells have actually invaded into other parts and they're forming secondary tumors. So this is where maybe like a breast cancer cells have gone into either the lymphatic system or into the circulatory system and moved into another area, and they've metastasized into like the liver. So we know that a single mutation is not enough to cause cancer. If we look at individuals that are older, they have a higher incidence of cancer because their cells have gone over a number of rounds of division, and that as you get older, the number of mutations build up. And so as we can see in this graph, if we look at the incidence of colon cancer in women in England and Wales, we see as individuals age, the incident rate increases for colon cancer. So someone who is 20 years old is less likely to get colon cancer versus someone who is 60 or 70, right? And so keeping in mind that someone who's older, their cells have gone through multiple rounds of cell division. And so with each round of cell division, there's a potential for mutations to occur, okay? And usually what happens is through the steps of regulation that we talked about with cell division, those mutations would be caught and corrected, or the cell would be induced to go into apoptosis. And as we just mentioned, cancer is an imbalance of cell division, so many rounds of cell division, and so as a cancer cell is dividing and dividing and dividing, it's more likely to undergo mutations than the surrounding non-cancerous tissue. Also, we're not getting apoptosis. There's a loss of apoptosis. So cells that normally through regulation would be told to go into apoptosis and to die, they don't get told that. So when mutations happen to genes that relate to cell division or relate to apoptosis, those would be more likely to be lead to cancer than some other random gene that has nothing to do with those pathways, right? And again, because there is so many steps of regulation, it's not that one gene, one mutation is enough. We would have to have multiple genes mutated, okay? And as we age, as we have undergone more rounds of cell division, there's more likelihood that those mutations have occurred, okay? And so really, cancer is a rare thing to occur. It happens infrequently because we have so many steps of regulation. So not to diminish anyone who knows someone who has cancer or has experienced cancer, it does happen. But just like development, the stages of development, for all of us to get here, a lot of things had to happen correctly. And so, and cancer occurs when multiple things happen incorrectly. So this figure here looks at a karyotype, and so the karyotype on the left is of a typical cancer cell. The karyotype on B is a specific type of cancer, and so for this colon rectal cancer, there's actually defects in the DNA mismatching repair genes, so genes that would allow you to realize that nucleotides have been incorrectly put into locations and then be repaired. Okay, so we can see with both of these cancers that these are not your typical karyotypes. So we see that there are in our one on panel A, we see that for some of these chromosomes, we have multiple chromosomes present that should not be there. So additional chromosomes indicating that cells did not separate correctly. And also that checkpoint of are there the correct number of chromosomes in each cell before we undergo cytokinesis did not happen. So really when we look at cancer, one of the hallmarks is genetic instability, right? So these karyotypes do not look like normal karyotypes. The genome has not been maintained. So for most cancer, they derive from a single abnormal cell. So your tissues are made up of millions of cells, and one cell ends up having a mutation, multiple mutations in it. And that cell then, because of the mutations that have occurred in cell division and rapid cell division, it begins to increase in numbers, and to the point that you end up getting a tumor being formed. So if we look at tumor progression, it really is kind of a process of natural selection, right? So we have this accidental production of a mutant cell, as we can see on the figure on the right. And that cell doesn't go into apoptosis. It's allowed to survive. Now we end up having a second mutation occur. Again, that cell doesn't go into apoptosis, it's allowed to divide, and then another mutation occurs, and so then there becomes a third mutation. And with each one of these mutations, that regulation, that telling the cell to go into apoptosis does not occur. The cell is allowed to survive, again, because there's been a break in the normal regulation of the cell cycle. It's going to be rapidly growing and dividing, and so it's going to end up taking over that area and become very invasive. So when we look at genes that lead to cancer, they can either be due to a gain of function or a loss of function. Okay, so for oncogenes, what's happened is that we have a mutation to the oncogene to the gene, and because of the mutation to the gene, it actually promotes cell transformation, cell division, and so that then lends it to be likely to developing cancer. And so again, with oncogenes, it's a gain of function, an increase in activity. With tumor suppressor genes, what happens is that we actually need both genes to be mutated. So with tumor suppressor genes, those are genes that suppress tumor activity. And so these would be pro-apoptotic genes. And so because we have two copies of our genes, because we got one from mom and one from dad, we actually would need both of these tumor suppressor genes to be mutated and inactive. And so if those are inactive, we're no longer able to suppress with that gene. And so that could promote cell transformation, promote cancer. Okay. So oncogenes usually are identified because we can add that gene into like a model system. And so if we add the oncogene, then the cell's phenotype, how it behaves, changes, and cell division increases. Where tumor suppressors, we'd actually need to remove and delete both copies of that in order to study that. So the upregulation of proto-oncogenes or oncogene expression can occur through different mechanisms, and so this figure outlays those different mechanisms. So we could have this oncogene where we get this deletion or point mutation in the coding sequence, and so this actually leads to a hyperactive protein being made. So it's this, the levels are staying the same, it's just that protein is overactive, it's able to bind more strongly. We can also have a mutation in the regulatory region, and so if there is a dysregulation or a lack of regulation and that the gene would always be turned on, we're going to get overproduction of the protein. Okay. We can also get gene amplification, so where the gene is replicated in multiple locations, so turning the gene on actually gets multiple copies of the protein because we have multiple copies of the gene, so we get this overproduction in that way. We can also get chromosomal rearrangement so that the gene is found on a different chromosome, and because of that, promoter regions and such could be changed, and so we get an overproduction of the protein because of that, because of the change of the chromosome. So certain chromosomes are going to be turned on more frequently than other chromosomes, and so if the chromosome is being opened and utilized for other genes, it can then make it more likely for that gene to be turned on and protein to be made and get overproduction. We can also get this depending upon where the gene has been inserted, we can get a fusion protein that develops, and that would be a fusion. A part of that protein is from the gene that normally is on that chromosome, and then our proto-oncogene. So tumor formation requires two steps. There's the initiation and then the promotion. So we're going to have some kind of DNA damage done through an initiator. So this could be exposure to drugs like carcinogens or X-rays or UV light. So we have the damage, and the damage is not enough, right? We have to have that cell survive so that it is able to grow and divide. It doesn't go into apoptosis. So usually then there is a promoter. There's exposure to another agent that's actually going to promote cell division, or exposure to inflammatory pathways that allows for that cell to kind of go unchecked and continue and divide. So normally, if that cell's just by itself, even though it has that mutation, keep in mind that, just like we saw in with the cell division and in culture, neighboring cells are going to inhibit the growth of others because there's only so much growth factor, right? And so normally, even though we have this cell, its growth is going to be restrained, but something happens, exposure again to an agent or inflammation, that will actually promote this tumor cell, this precancerous cell, to then grow and divide and expand in its population, okay? And as it expands, then there's an increase, because of this increased amount of cell division, there's an increased probability that it'll acquire a second mutation for it. So if we look at the stages of tumor development and mechanism of metastasis, we'll recognize that there's a number of stages, which we've kind of alluded to already. So we're going to have this genetically altered epithelial cell, and so not genetically altered in the sense that, someone in the lab genetically altered it, but that there's been some kind of mutation that has arisen that causes their genome to have changed and become unstable. And then we'll get this hyperplasia, where the cells begin to divide rapidly. And so again, this on-check of cell division. Then we'll get this dysplasia, where the cells actually start to change in their form. So they stop appearing like the neighboring cells. And so for histology purposes, this is where you could start to see changes, visual changes in the cells. Then in stage C, we have our in situ cancer. So this is where the tumor is in place, so benign. It's staying in that location, but again, we have this clear definition of the tumor. It's not invading the surrounding tissue, but it's definitely not the same as the other tissue around it. And then we have a malignant tumor, which is what we'd traditionally call cancerous. This is where it's invading the normal tissue. It's entered into the bloodstream or lymph node, okay? And so this is affecting the function of the neighboring tissue around it because of that increased growth present. And then lastly, this is where we can have invasion out of the original site of the tumor through the bloodstream or the lymphatic system, where we would get metastasis into different locations. So just keep in mind that for metastasis, it's not that it's just moving to the next neighboring side. It could be moving to a totally different organ system. And again, this is where CT scans are usually done to screen to see whether there's metastasis to different location, and then they would then be treated. So there are a number of critical genes that, if mutated, can lead to cancer progression. So obviously ones that relate to cell growth, cell division, apoptosis, DNA repair, genome stability, so separating of those chromosomes, Angiogenesis is also really important. So angiogenesis is the formation of blood vessels. And so as the tumor grows, it needs to have a blood supply, or the tissue becomes necrotic, so it can start to ulcer. So in some cases, when you have a benign tumor, if it grows large enough and it's not promoting angiogenesis, it will actually start to collapse in and on itself. And start to ulcer, because it's not going into apoptosis, it's going into necrosis, because there's not enough oxygen and nutrients to support it. Okay. Genes related to metabolism can lead to cancer, and also extracellular matrix, and there's also epigenetic alterations. So epigenetics is like methylation, putting methylene groups onto genes to turn them off or turn them on. There's other epigenetic markers than just methylation, but this can change the gene that's actually being utilized. And so normally our cells are actually pretty good that if there's a mutation that makes a gene ineffective, you have 2 chromosomes, it'll use the other chromosome. And so epigenetics allows for the best chromosome to be used, but if that gets altered, then you're using a less effective gene. So very interesting area of research. So these are just some of genetic abnormalities related to colon rectal cancer. So you can see some of these are oncogenes involved in signaling pathways, where other of these are tumor suppressor genes. And you can see TGF-beta is related to inflammation, response to stress, and DNA damage. Okay, so. And then lastly, I just want to show that there are a number of different types of cancers. So they're usually classified according to the tissue type that they're in effect. So carcinomas are usually epithelial tissue. Sarcomas would be muscle or connective tissue. Leukemias would be white blood cells. So blood cancers. Lymphomas would also be the white blood cells and precursors. Gliomas are the glial cells in the central nervous system. So if you look at the type of cancer, sometimes the name will tell you a little bit about the cancer. So obviously, if someone has a cancer and it's been found in a location that is not where it has arisen, it would indicate there's a metastasis. So lastly, just to look at 2 more specific types of cancer, basal cell carcinoma. So these are actually derived from keratinocytes. So they oversynthesize cytokeratin intermediate filaments, and usually are only locally invasive, and they rarely metastasize. Melanomas are derived from the cells that produce pigment in our skin. So often they will continue to produce a large amounts of the pigment, so they're going to have a different colorization than normal. They are highly invasive and highly likely to metastasize. So you'll end up with these melanocytes, these pigment cells in different locations, and they continue to produce the pigments in these different locations. So they're relatively easy to identify as a metastasis in these different locations, because you wouldn't normally have melanocytes in your liver, right? So this concludes the slides on the stem cells and cancer. And so as always, if you have any questions related to chapter 20, material that's covered or not covered from the textbook, please contact me, and I'm more than happy to go over that, definitely during class time or outside time. All right, thank you.